Good morning, friends. So, I welcome you all again. So, we were talking about the cell organelles. So, in today's lecture, we'll be going to start with the ribosomes. Okay, so in this video, we'll be going to discuss the structure of ribosomes and the functions of ribosomes, and as well as the biogenesis of ribosomes. So, these ribosomes, uh, actually, these are not organelles. So, because they don't have their own membrane. So ribosomes are actually structures which are made up of from the from the RNA and proteins. So these are a specialized structure which are made for the synthesis of proteins. So these ribosomes actually first described by the GE Palade. So the ribosomes occur both in prokaryotic and eukaryotic cells. So these ribosomes are actually found in both prokaryotic as well as in eukaryotic cells. So in case of prokaryotic cells, when we talk about these prokaryotic cells or prokaryotic organisms, these ribosomes are found in freely in the cytoplasm. So there is no definite nucleus. So when the DNA of the prokaryotic cell produces RNA RNA by the process of transcription, then these ribosomes, which are freely present in the cytoplasm, produces the or synthesizes the proteins with the help of process of translation. So in case of eukaryotic cells, these ribosomes are actually some ribosomes are freely in the cytoplasm and some are attached with the endoplasmic reticulum, that is called as rough endoplasmic reticulum. So the transcription process in case of eukaryotic cells is takes place inside the nucleus. So after transcription there is a production of RNA, and these RNA then later on transfer to the cytosol of the cell, and in the cytosol, then in eukaryotic cell, the synthesis of proteins is takes place, that is the translation process is present in the uh, cytosol. So two types of ribosome have been recognized, that is 70s and 80s type. So there are two types of ribosomes, that is 70s and 80s ribosomes. So these es stand for Swedberg unit, or the it is also called as a sedimentation coefficient. So when we isolate ribosomes from the cell, with on the basis of that, these units are actually given to the ribosomes and ATS type of ribosome. So this type of ribosome is found in the eukaryotic cell, and this type of ribosome is found in the prokaryotic cell. So the S is Swedberg unit, that is sedimentation coefficient, and is related with the size and molecular weight of the ribosome. So when we isolate ribosomes with the with the help of this size and molecular weight, so these unit actually is related to this size and molecular weight of the ribosome. So remember these points, that what is actually 70s and what is 80s and why it is so called. So the 70s ribosome occur in the prokaryotic cells of the blue green algae, then bacteria. Okay, so these 70s ribosomes are present in blue green algae, then in bacteria, and also in mitochondria and chloroplast of eukaryotic cells. So you must remember these points, that actually where these ribosomes, that is 70s ribosome, are actually found in the living cells or living organisms. So the 70s ribosomes found in blue green algae, then in bacteria, and as well as in the mitochondria and chloroplast of eukaryotic cells. So in case of mitochondria, the ribosomes may be found like 70 years, then 77 years. So on the basis of this sedimentation coefficient, there are different types of ribosomes in mitochondria. So the ATS ribosome occur in eukaryotic cells of the plants and animals. So primarily these ATS ribosomes are found in eukaryotic cells of the plants and animals. So this is the general information about these ribosomes and the different types that which are found in living cells. So let's see the structure of 70s ribosome. So the 70s ribosome consists of two subunits. Okay, so again the 70s ribosome is divided into two subunits, that is 50s and 30s. Okay, so when we isolate these entire prokaryotic ribosome, that is 70s, then it is called as a 70s ribosome. But when we separate these two subunits, that is a larger subunit and smaller subunit, then it appears as a 30s and 50s. So the smaller one is 30s and larger one is 50s subunit. So the 50s ribosomal subunit is larger in size and has the size of 160 angstrom to 180 angstrom. Then the 30th ribosomal subunit is smaller in size and occurs above the 50th subunit like a cap. Okay, so this is larger subunit and this is smaller subunit. So smaller subunit is actually uh, forms a cap on this larger subunit. So the two ribosomal subunit remain attached with each other due to high concentration of the Mg plus Mg2 plus ions. So these two subunits are actually attached with the help of this Mg2 plus ions. So these points you must remember. So when there is an absence or a low concentration of Mg plus plus ions, then you will find these subunits separate or apart from each other. So this is the points that you must remember. So now let's see the ATS ribosome. So the ATS ribosome also consists of two subunits, that is 60s and 40s subunit. So the 60s subunit is a dome shaped and larger in size, again as in the case of 70s ribosome. Again these a larger subunit, that is the 60th subunit of 80s ribosome is larger in size. The 60th ribosome remain attached with the endoplasmic reticulum, as I told you. The 40th ribosomal subunit is smaller in size and occurs above the 60th subunit, forming cap-like structure. So in case of eukaryotic cell, these 80th ribosome actually divided into two subunits, that is 60th subunit and 40th subunits. So the okay, so this is a smaller in size and forms a cap-like structure. So both the subunit remain separated by a narrow cleft. So when these two subunits attached to each other with the help of Mg plus plus ions, these two subunits are actually attached with the help of Mg plus plus ions, and when they attached, they actually forms a narrow cleft. These narrow cleft are actually for the sitting of the RNA molecules. So this is about the ATS ribosomes. So okay, so this is the ribosomal structure which are actually described by the Stoffler and Whitman's model of 70s ribosome. So uh, this structure, if you'll see the structure, this is the larger subunit, and that is dome shaped. So this is the base. This is 50th subunit of a 70th ribosome. Then this is the cleft, and that is a narrow cleft or space which are actually remain open for the sitting of RNA molecules when there is a process of translation or the synthesis of protein molecules takes place. So this is the head. Then this is central protuberance. So this is the body and this is the 30th subunit. So this ribosomal structure actually or the model is actually put forth by the Stoffler and Whitman's, that is 70th ribosome. So here again we have another model of 70th ribosome which are put forth by the or which is also called as Lex model of the 70th ribosome. So this is a three-dimensional structure which is uh, actually uh, put forth by this Lex model or Lex scientist. So this is the smaller subunit, this is the larger subunit, and after unite, so this is the complete ribosome, that is 70s ribosome. So again same, this is a smaller subunit, this is larger subunit. So this is the head, then cleft, then this is the stalk of larger subunit, this is the valley ridge, and here is the three-dimensional structure. And again you can see that this is the narrow cleft which is present here for the sitting of RNA molecules. So these are two different models of 70s ribosome. So this is this is the process how translation, this is the translation process how it takes place. So this is our ribosome, that is three-dimensional structure. So uh, from this narrow cleft there is a, so this is our RNA molecules, and this is narrow cleft, which are actually used for the uh, attachment of the amino acids for the synthesis of protein. So this is the messenger RNA which are situated inside this narrow cleft. So this is the load. This is 40s subunit. So okay, so this is the eukaryotic ribosome. So eukaryotic ribosome contains 40s subunit and 60s subunit. So this is the endoplasmic reticulum. So on the cell membrane of this endoplasm, on the uh, plasma membrane of this endoplasmic reticulum, there is a ribosome which are attached to this membrane. So this is the messenger RNA which are actually fitted into this narrow cleft of this ribosome, and by reading this information of the messenger RNA, this ribosome reads the message from this RNA, and then the attachment of the amino acids with the help of peptide bond takes place, and the synthesis of this protein is actually taking place. So the amino acids are actually bring to the ribosome with the help of this tRNA molecule. So there are different components which are involved in the process of protein synthesis. So this is the general process of protein synthesis, or the structure of ribosomes which are involved in the protein synthesis. So here you can see the molecular weight of the 70th ribosome, that is 2.5 million. Then this is the eukaryotic ribosome of a molecular weight of 4.2 million, that is double to that of 70th ribosome. So when we separate the 70th ribosome as a 50th subunit and 30th subunit, then this 50th subunit have a molecular weight of 1.6 million, and this is the molecular weight of 900,000. So this is the molecular weight of this 50th and 30th components. So again, when we separate, or when we isolate, or we when we see the chemical components of these 50th subunit, we will found that there is a different rRNA molecules, that is ribosomal RNA molecules. So we have 5s ribosomal RNA, then we have 23s ribosomal RNA. So you should remember these points, that 5 years are RNA, 23 years are RNA are found in this 50th ribosomal subunit of the 70th ribosome, and which are present in prokaryotic cells. So this 50 and this larger subunit of ribosome contains 5 years ribosomal RNA, then 23 years ribosomal RNA, and these uh, rRNA, when it is of 2900 nucleotides, and this rRNA is of 120 nucleotides. Then this is the content of ribosomal RNA, but these subunit is actually is again contains some protein molecules, that is 34 protein molecules. So together these 5 years ribosomal RNA, 23 years ribosomal RNA, and 34 proteins constitute these 50 years ribosomal subunit. Okay, so like that, these 30th smaller subunit of these 70th ribosome contains the 16 years ribosomal rRNA. So this is the 16 years rRNA, which actually contains the 1540 nucleotides, and it also contains the 21 proteins. So uh, again, the another point, that is these rRNA, that is 16 years rRNA, these ribosomal RNA is restricted to the prokaryotic cell, or it is restricted to the bacterial cell. So the 16 years ribosomal RNA uh, rRNA is only found in the prokaryotic cells, or only found in the bacterial cells. Okay, so this is the point, and then that is the point you must remember. So this is again the eukaryotic ribosome. When we isolate, or when we isolate this, it is ribosome, we can found that, and there is a large subunit, that is 60s, and smaller subunit, that is 40s ribosome. So the molecular weight of larger one is 2.8 million, and smaller one is 1.4 million. So again, this larger subunit of 60s ribosome contains the 5s rRNA, 28 years rRNA, and 5.8 years rRNA. So the first RNA contains 120 nucleotides. These 20S RNA contains 4700 nucleotides, and these 5.8 years rRNA contains 160 nucleotides. Okay, so together all these three rRNA molecules contains the 45 proteins. and when these four different components constitute together forms a 60s ribosomal subunit. So this 40s smaller ribosomal subunit contains the 18 years ribosomal RNA. So these 18 years ribosomal RNA, when combined with these 30 proteins, it forms the 40s ribosome, that is a smaller subunit of 80s ribosome. So this is the chemical content which are present in the ribosome. So this is a very important point that you must remember. So okay, now let's see the function of ribosomes. So ribosomes play a very significant role during the biosynthesis of protein. So this is the very important and very primarily important function of the ribosomes, that is the biosynthesis of proteins. So ribosomes provide the safe and suitable facility for the synthesis of proteins. So ribosomes also provide the safe and suitable facility for the synthesis of ribosomes, or the synthesis of proteins. So ribosomes translate information from mRNA, that is messenger RNA, with high degree of accuracy. So when the protein synthesis process is actually going on, so the degree of accuracy is very high, is just because of these ribosomes, because the ribosomes read the exact message of these mRNA molecules, and then only the information provided by this mRNA is actually used to bring the amino acid, and then these amino acids are joined together to form a protein. So as ribosome are the only site of protein synthesis, they are called as factory of protein synthesis. So these ribosomes are also called as factory of protein synthesis. So there are many more functions of these ribosomes, but uh, I just mentioned few of them which are very important, or that you must remember. So the next point, that is biogenesis of a ribosome. So this point will be going to discuss in the next video lecture. So till then, just watch the previous video lectures and keep watching and keep supporting. Thank you.